Welcome to this lecture on microbial ecology and environmental biotechnology part C, and in this particular lecture we are going to discuss on the principle of good management with reference to the application of the knowledge and understanding of microbial communities in various fields of environmental biotechnology. Now, the most fundamental question at the interface of microbial ecology and environmental biotechnology is discussed, and this particular question is very prominent, and the question is how do microbial communities self assemble to achieve and maintain a function. Because whether the goal of utilizing this particular concept of microbial ecology into environmental biotechnology is basically gaining scientific understanding or applying that understanding where microorganisms are functioning and functioning towards production of certain compounds or cleaning up certain waste materials or certain other purposes. So, applying that understanding and sometimes to create a high value services or products, in both the cases we need to have an answer to this question, that how do these communities which occupy or work in a particular environment and a particular system where these environmental biotechnology related processes are going on, they self assemble and they function. Now, environmental biotechnologists depend on self assembly in the sense the self assembly of different species once the proper conditions are in place, and self assembly of these organisms need to be based on identifiable principles that can be put into practice. And the overarching question then leads to certain corollary questions, that what are the underlying principles of self assembly, because the assembly must be guided by certain factors, certain processes. So, who are those? What are those factors? How the microbial species are actually involved in a synergistic or in a kind of antagonistic assembly, because often in a community which is basically used for any particular process, we find that there are numerous species which are forming different type of clusters. Those clusters are basically the assembled species members. So, these assembled species member forming the clusters could be positively correlated, that means they must be having some synergistic relationship, or they could also be having some kind of negative interaction between themselves and basically developing antagonistic or portraying the antagonistic relationship among themselves. So, taking into account all these synergistic as well as antagonistic reactions, we can very well understand the process of self assembly, but we need to, before we understand the self assembly, we need to first define what are the principles of self assembly. The second question would be that what community structure is optimal for a biotechnological application. So, whatever may be the applications, it may be the production of biofuel, it may be production of methane, it may be decontamination of certain waste material. So, whatever it is, so we need to actually understand that what community structure is optimal for a biotechnological applications. The next question is what environmental conditions trigger assembly of the desired community with its desired function. This particular question is very relevant, because we know that in a particular community there could be several hundreds or even thousand or more than thousands microbial species, and these species are expected to form different kind of self assembly or clusters, and these clusters, as I said, could be synergistic cluster, could be antagonistic clusters. So, some of these organisms within the clusters, or some of the clusters in fact they might be useful for the desired community function. Now, what environmental conditions will trigger the appropriate assembly formation, that is the particular assembly or particular cluster which is required or optimal for a particular community function would certainly be dependent on certain environmental factors. So, what are those environmental factors which triggers the or trigger the assembly of the desired community function that has to be delineated. And the next and the last question in this case is, are the conditions and outcome are predictable, reproducible and controllable. So, the conditions which actually guide this assembly, particularly the assembly which allows the desired community function, are these conditions which actually favor the formation of those kind of clusters or those kind of interactions are predictable, and we can reproduce them and we can control them. And it is not only the conditions and also the outcome, because the desirable condition will only lead to the appropriate outcome. So, once we are able to control the desirable conditions, it is expected that the outcomes are also going to be controlled. So, one of the major questions in this case is basically these conditions and outcome, which basically are defining the desired community formation, community assembly with respect to a particular environmental function. Are they predictable, reproducible and controllable? So, in most environments, microbial interactions take place within microscale cell aggregates. As I mentioned, there are synergistic interactions and there are antagonistic interactions. So, often we found that these interactions are taking place in a microscale cell aggregates. Now, at the scale of these aggregates, that is around 100 micrometer, interactions are likely to be dominant driver in population structure and dynamics. And in a microbial ecology study with respect to its application in environmental biotechnology or for basic understanding of microbial ecology function, understanding these interactions are one of the priority areas. Now, in particular, organisms that exploit interspecific interactions to increase ecological performance often co-aggregate. So, this is basically the synergistic interaction. So, there are, as I said, there are many species. Out of many species, some species would be engaging with interactions between themselves, which will be helping them to perform better, to utilize the resources there, and also might be useful for producing or functioning to the extent that is desirable to us. Us means the environmental biotechnologist. So, some organisms will be exploiting the intraspecific interactions which are synergistic and which will allow them to co-aggregate, that is, they will form distinct clusters. Conversely, there would be some organisms who are not involved in the previous cluster, and rather they might have some antagonistic role with respect to each other or with respect to other cluster, and they will tend to form separate clusters often, or they may remain isolated as discrete members. Often very often they form different clusters, and that might create distinct sub communities or sub clusters and increase diversity at larger length scale. So, if we look at those communities at a higher scale, we will find out that there are actually more diversity than number of species present. So, multiscale structure of the system and their arrangements are very important. So, one of the fundamental properties of complex system are the multiscale structure, and ecosystems have a hierarchical arrangement which might go from the meta communities to communities, populations, individual pathways and genes. For example, if we look at these different communities which may be expected in different environment, we will find out that in a length scale the drivers are entirely different. So, in a scale of like centimeter to meter, we may have environmental gradients which are controlling some meta communities or meta populations, whereas at the scale of 10 to 10 to the power 3 micron or so, we can have local communities or populations. We will have like a biofilm frogs or a kind of cell, different cells are growing within a community scaffold. These are all multi-species aggregates, but when we come down to micron level or micrometer level resolution, we will be able to find out the single cell physiology, and we will be able to find out that there are actually discrete sub clusters or sub aggregates are there. So, those are basically synergy, reflecting the synergistic and antagonistic interactions among the members. So, self assembly, which basically enables us or provides us information about all these micro details about how the community members are structured within a particular system and what factors control them. Particularly a particular cluster, a particular assembly might be very critical for a particular function. So, knowing that, defining that and predicting their behavior is a very important task with respect to translating the knowledge of microbial community into environmental biotechnology. Now, self assembly also implies that microbial communities behave as a kind of multicellular organisms, because all these microclusters or clusters, they are all tied together. The synergistic cluster, the antagonistic clusters, all these clusters are basically components of a particular community. There would be some connections which are already existing there, and these in a totality they behave like a multicellular organism. Now, as we understand that, some more questions along this line of inquiry include: does the community have a multicellular life of its own, and how do the microorganism in the communicate and coordinate to gain the advantages of a multicellular state? Why do they like to achieve this multicellular state with numerous or several clusters or interactive assembly or sub assemblies? They form this kind of multicellular structure. It is actually an entire structure like a biofilm. So, how they communicate and coordinate with themselves, that is a big question. And what are the advantages, if any, to the community and to the human? So, to the community in particular and to the human, where we are actually trying to explore or exploit the benefit out of this. So, what are the advantages of these kind of interactions between themselves? Now, the next point that we are going to discuss here is managing the microbial community, because essentially this particular aspect, managing the community, will enable us to gain the outcome from the community or the microbial members, because when we target that a particular community or a particular microbial system would be delivering some function. So, be it a treatment of wastewater, be it a production of certain compounds or increasing the certain other activities. In all the cases, it is basically managing the microbial community, and it has been said that it is actually a win-win situation. So, here we discuss this particular point of this win-win situation, why we are saying it is a win-win situation, through certain concept or principles of good management. Now, managing a microbial community means creating a technology that works for the microorganisms, so that they work for us. Just assume that we want to use an activated culture, activated sludge for wastewater treatment. So, if we understand how the organisms present within the activated sludge actually work with respect to degrading the degradable pollutants which are present in the wastewater, we would able to exploit them, apply them and get the benefit out of them in the best way. That is why it is said that works for the microorganisms, so that work for us. Now, here I would like to take one particular example other than the waste water treatment, that is activated sludge that I just mentioned, that enhancing the carbon dioxide fixation by autotrophic bacteria, because lot of interest is being paid on this particular aspect, owing to the rising concern over the atmospheric carbon dioxide level and its impact on the climate. So, there are defined strategies to enhance the CO2 fixation by autotrophic microorganisms. So, basically what is autotrophic carbon dioxide fixation? It is basically a pathway, like here it is depicted, that is a pathway which is driven by these autotrophic microorganisms, and it allows the cells to transform the inorganic carbon like HCO3 or CO2 into organic metabolites like acetyl CoA and pyruvate. So, autotrophic organisms are already existing, they are there in the environment. Now, the CO2 fixation by these autotrophic microorganisms can be reinforced by improving the efficacy or efficiency of the CO2 fixing pathway, by developing more energy harvesting system to improve the supply of ATP and reducing power, etc. So, let us see what are the ways to do that. Now, it can be done number 1 by engineering the CO2 fixation pathway. So, once we have certain good organisms which are candidate organisms, we aim to engineer the CO2 fixation pathway, and the efficiency of the native CO2 fixation pathway can be increased by overexpressing its key enzyme, like putting them into a plasmid vector and then overexpressing them, or sometime replacing them with a better enzyme, that is a part of the metabolic engineering, I will say. More efficient synthetic CO2 fixing pathway can be also designed using synthetic biology and other omics data. So, these are all open ended research areas where people are working on that. Similarly, the next option could be development of energy harvesting systems. Like efficiency can be improved also by optimizing the natural photosystems in natural autotrophic microorganisms, creating artificial photosystems for non-autotrophic microorganism, or developing some electricity utilizing system. So, where non-autotrophic organisms can be made to do carbon fixation, and essentially we can allow them to convert carbon dioxide to produce something like electricity utilizing system or something like that. The last one could be regulating the distribution of carbon flux. Now, the distribution of cellular carbon flux can be regulated through enhancing the product synthesis pathway, because flux is basically the rate of flow of metabolites. So, as inorganic carbon will flow through that, engineering the transcription factors and providing the organic carbon resources, etc., would be immensely beneficial. We have found that. Now, for all these things, for all this carbon fixation in autotrophic microorganism based system, which is one of the best areas or one of the most important areas of environmental biotechnology, we need to know how these actually autotrophic microorganisms which are present so abundantly in the natural environment, they actually function. They actually function in terms of their interaction with species member, their interaction with the physical and chemical materials that they use as a resources, or they could be affecting the microorganisms, and essentially also how their genes and how their metabolic functions are coordinated through these self assembly processes. Now, therefore, developing this technology that includes managing a microbial community works for the microorganisms, so that they work for us, is a win-win technology, win-win situation, because we need to know the microbes, their interaction, their function, how they work for a particular process very well. Then only we will be able to deploy them or apply them for our purpose. So, whether they will be useful for our purposes or not, that means whether they will work for us or not, it depends on whether that itself works for the microorganism. Now, creating that win-win technology requires that the knowledge gained from microbial ecology research be translated to a practical setting. And how it is accomplished? There are 5 principles. These 5 principles are: aim for the big benefits, develop and apply more powerful tools to understand the microbial communities, follow the electrons, retain the slow growing biomass, and integrate all the concepts, all the knowledge that we derive from these our studies on microbial community function, microbial community structure, their interaction with respect to any given environmental setup. So, with this this topic is ended here, and for this particular portion I would recommend these 2 following articles, that is the Microbial Ecology to Manage Processes in Environmental Biotechnology by Bruce Rittman, and a Vista for Microbial Ecology and Environmental Biotechnology by Rittman et al., published in Environmental Science and Technology in 2006. So, in conclusion, environmental biotechnologies depend on self assembly once the proper conditions are placed, and the multi-scale structure of the system and their arrangements are very important. Self assembly is also very important to understand for understanding the community function, and it also implies that microbial communities behave as a kind of multicellular organism. managing the microbial community essentially is a win-win situation, and we have 5 principles towards that. Thank you.